right, welcome. Ms. Tavolera here. Ms. Waldron's recording, so we're both here and we're ready to take you through photosynthesis. First part, we want to go through the equation for photosynthesis. So what are we starting off with? Well, we need some water. We need some carbon dioxide. And we also need some light. So these will be our reactants for photosynthesis. What are we going to produce? We'll produce oxygen and we'll produce glucose. So this is our formula for glucose. All right, so now what we're going to do is we're going to head over to our light reaction. This will be our first step for photosynthesis, so I'm going to write it out right here. We are in our light reaction. And from what it sounds like, we need to use sunlight for the light reaction. First thing we want to go over is this entire thing on the board is a chloroplast. That is where photosynthesis is going to take place. The different steps, or the two stages, take place in different areas within the chloroplast. So our first area that we're focusing on is actually going to be our thylakoid membrane. That's where our light reaction is going to take place. Before we can get into our thylakoid, we're in the stroma to begin with. The stroma is like this liquid, fluid area that's in there. Water is going to be absorbed through the stomata. And what's going to happen is light is going to hit this water, and it's going to split it into three things. It's going to split it into hydrogen, oxygen, and an electron. This oxygen is then going to be released out of the chloroplast, and it's going to be exposed to the atmosphere, which is what we are going to breathe in. So that's the byproduct of photosynthesis, will be oxygen. And we can also see it in the equation. Oxygen is a product. All right, so now we want to focus on this electron. This electron is going to go into our photosystem 2, and the only reason why it's called photosystem 2 is because it was discovered second. Photosystem 1 was discovered first, so they named it photosystem 1. All right, so the electron goes into our photosystem 2, doesn't have that much energy, so how do we excite our electrons? We got to hit it with some sunlight. So sunlight hits our photosystem 2. And the reason why it's going to actually be attracted to our photosystem 2 is because it has chlorophyll in it. So this is our light absorbing pigment. That's why we have it in green, if you can see. It's going to attract the sunlight. All right, so that electron that's in here is going to get really excited. It's going to start bouncing around from chlorophyll to chlorophyll. Okay, here we go, it's bouncing until finally it's captured up here. The full name of that, which is the primary electron acceptor, is what's going to hold on to it. And now we know this electron is still very unstable. So what's going to happen? The electron needs to lose some of that energy. So it's going to move down this ladder, and as it's moving down the ladder, it's going to be releasing energy, and hydrogens that came from our water, right, so hydrogen split off, hydrogen will be pumped into our thylakoid. So hydrogen needs this energy from our electron moving down the ladder so that it can be pumped into our thylakoid. All right. Next thing that's going to happen is this electron that just moved down the ladder will go into our photosystem 1. Now this electron doesn't have so much energy anymore, so our light will come along, hit the electron becomes excited, starts bouncing around, until finally it's going to be captured by our primary electron acceptor. Okay, that electron, once again, very unstable. And how do we become more stable? We've got to move down our ladder. So we're moving down our ladder. Anytime an electron is moving down this ladder, it is releasing energy, right? also, that electron is then going to help us reduce NADP plus to NADPH. So this process that just happened, we gained an electron, so we call this a reduction. We've just reduced NADP plus to NADPH. It got this electron from here, and now we have one of our energy sources for our Calvin cycle. All right, almost done with our light reaction. We got to deal with these hydrogens really quick. We're going to have a very large buildup of hydrogens in here. Usually what things want to do, they want to move from a high concentration to a low concentration. So these hydrogens are going to move through this device right here, which is called ATP synthase. So hydrogen will move through the ATP synthase, and as it's moving through the ATP synthase, ADP will bind with the phosphate group that's just kind of in here to become ATP. All right, so now we've made two energy sources, our ATP and NADPH. Where do we go next? To the Calvin cycle. So now we're moving on to our Calvin cycle because we have our necessary energy to power it. Okay, so now we're in our Calvin cycle. So the light reaction happened in the thylakoid. Calvin cycle is going to happen in the stroma. Stromas are a fluid, liquidy area of our chloroplast. All right, so we need to talk about a few starting materials. We are going to start with a molecule known as RUBP. What you really need to know about RUBP is a 5-carbon molecule, and that's really it, right? So here's our 5 carbons, 1, 2, 3, 4, 5, so that's 5 right there. And we also need something else. From our equation, we need carbon dioxide. Carbon dioxide will now go into our Calvin cycle. What's going to happen is this RUBP is going to bind with our carbon dioxide, but it needs a special enzyme to do this. And that enzyme is called rubisco. So rubisco will be the enzyme to bind RUBP and carbon dioxide together. That's going to form a 6-carbon molecule. So I label that as a 6-carbon molecule right there. It's very unstable. It splits almost immediately. That forms and splits into a PGA molecule. So a PGA molecule is a 3-carbon molecule. So we originally had 6, it splits into 2, and now we have 2 PGA molecules, right? All right, so now what's going to happen is this PGA needs to be rearranged a little bit. So in order to rearrange something, we need energy. We got these energy sources, ATP and NADPH, from our light reaction. So now they're actually being used. We're taking this ATP, we're taking this NADPH, bring it over to our Calvin cycle, and we're going to actually use them now. So ATP becomes ADP, NADPH becomes NADP+. So this reaction now is going to be called an oxidation. So I'll write that right over here. Oxidation is when we are losing electrons, and that's what just happened right here. Now we're going to go from our PGA. We rearranged it a little bit, and now we have our G3P molecule. And we made a lot of these G3P molecules. One of those G3P molecules comes out, so only one comes out, and a couple more are going to go. The rest of them are going to go back into our cycle. And the reason why we need that G3P here to go back into our cycle is so that we can then come back to our original starting material, which is our RUBP. So we have this G3P molecule, we have a whole bunch of them, we rearrange it a little bit, we need some energy to rearrange it, so we go from ATP, we're going to expend it, use it, and it becomes ADP. And now we're back to RUBP, and we can do the cycle again, which is what we want. I just want to point out that this cycle, the Calvin cycle, this G3P only is a 3-carbon molecule. When we look back to our equation, glucose is a 6-carbon molecule, which means that we need to have 2 G3Ps to make 1 glucose molecule. So that means this cycle has to happen twice. So we want to do the cycle one more time. We form another G3P, so 1 G3P plus another G3P will give us our final product of photosynthesis. And I hope that was helpful.